Welcome back to Dr. Liu's VCE Biology. Today's topic is the relationship between nucleic acids and proteins, as presented in the 2024 NHT Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. NADH, RNA, and ATP are not proteins, whereas DNA polymerase is a multipolypeptide protein, making option A correct. Each component of the TRP operon has a distinct role. TRPR produces the repressor protein, the promoter serves as the RNA polymerase binding site, the operator interacts with the repressor protein, and TRPE encodes an enzyme crucial for synthesizing tryptophan. As a classic example of repressible negative gene regulation, the TRP operon's repressor protein actively binds the operator to halt transcription when tryptophan is abundant. This is its primary negative feedback loop. When tryptophan levels are low, the repressor is released, permitting transcription to occur. Beyond this initial control, the TRP operon employs attenuation as a second layer of negative feedback control over its expression, further fine-tuning the process. Universality is a key characteristic of the genetic code, as identical codons, triplets, consistently specify the same amino acids across all forms of life. Furthermore, the code exhibits degeneracy, meaning that multiple distinct codons can encode the same amino acid. Translation, a fundamental step in producing proteins, relies on the coordinated efforts of three nucleic acid molecules. mRNA contributes the sequence information for protein synthesis, while tRNA is responsible for delivering the appropriate amino acids to the growing polypeptide chain. Lastly, RNA plays a multifaceted role by forming the ribosome structure, facilitating mRNA-tRNA interactions, and catalyzing the formation of peptide bonds. Organelle A, the Golgi apparatus, or Golgi body, is responsible for modifying and packaging proteins into vesicles. These vesicles then transport the proteins to various cellular destinations, including the cell membrane for secretion. This section on the relationship between nucleic acids and proteins is worth 11 marks, or 9.2% of the total 120 marks. Multiple choice questions, 3 marks. Question 1 tests knowledge of molecules made of polypeptide chains, such as DNA polymerase, NADH, RNA, and ATP. Question 2 focuses on the TRP operon, which regulates tryptophan production in bacterial cells, and the roles of its different regions. Question 3 asks for the differences between the attenuation and repression mechanisms in tryptophan production. Repression involves a protein that blocks transcription initiation, while attenuation regulates transcription termination. Short answer questions, 8 marks. Question 1a requires you to explain the features of the genetic code, specifically why it is both universal, consistent across all life, and degenerate, multiple codons code for the same amino acid. Question 1b asks for a description of the steps of protein production in a cell, focusing on the process of translation and the specific functions of mRNA, tRNA, and RNA. Question 1c requires you to explain the Golgi apparatus's role in modifying and packaging proteins for export. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.